Welcome back to the deep dive. Today, we are plunging into the microscopic world of cancer, and we're focusing on a very specific battlefield.
That's right, the tumor immune microenvironment, or what researchers call TIME.
And this isn't just the tumor itself, right? It's much more complex.
Oh, absolutely. It's a whole ecosystem. You have the cancer cells, of course, but also all the different immune cells, T cells, B cells, macrophages, plus the cells that give it structure, like fibroblasts.
And understanding not just who is in that ecosystem, but where they are. That's the critical piece.
It's everything. Their spatial relationship is key to predicting how a cancer will progress and, more importantly, if a powerful treatment like immunotherapy is actually going to work for you.
But getting that spatial information, especially at scale, has always been the big hurdle. We started with the old workhorse, immunohistochemistry, IHC.
Right, and think of IHC as just a single static snapshot. It uses a stain to light up one specific protein.
Just
one per tissue slice. So if you want to understand this complex ecosystem with dozens of players, a single snapshot is just completely insufficient.
So we evolved. We got to what is now the gold standard, multiplex immunofluorescence, or IM.
And IM is incredibly powerful. Instead of one protein, you can stain for, 21 or even more, all on the very same piece of tissue.
So you get to see the whole dance.
You see the whole dance. You preserve that crucial spatial architecture. But, and this is a huge but, it comes with a massive trade-off.
It's expensive.
Incredibly expensive, slow, you need specialized labs, and so the data is just scarce. We simply can't do the kind of population scale research we need with it.
And that scarcity is the exact problem we're digging into today. The solution we're looking at is a multimodal AI framework called GigaTIME.
Yes, and GigaTIME is designed to build a bridge between the chief abundant data we do have and that high-value data we so desperately need.
The abundant data being the standard pathology slides, hematoxylin and eosin, or H&E.
Exactly. The pink and purple slides every hospital has archives full of. They show us cell morphology, the shape, the structure, but they don't explicitly show us the protein activity, the cell's actual state.
So GigaTIME learns to connect the two. It learns how the subtle shapes in H&E predict the protein signals you'd see in MyY.
It basically teaches the AI to infer, or you could even say hallucinate, that missing MIF data.
And the scale of this project is what really stands out. This isn't some small pilot study.
Not at all. The sources show GigaTIME was trained on a dataset of 40 million cells with perfectly paired H&E and IMF data across 21 different proteins. That's the engine.
An engine that was then deployed on a massive real-world population.
A monumental population. We're talking 14,256 patients from 51 hospitals across 7 U.S. states. He covers 24 cancer types and over 300 subtypes.
So what was the final output of a calculation like that?
Almost 300,000 virtual MI slides. The mission here is to create a clinical atlas to unlock discoveries at a scale that was just impossible before.
Okay, let's break that down. The challenge is time's complexity, which needs multiplexed MIF imaging, but that's too rare and expensive.
And the solution is GigaTIME, which uses AI to translate cheap, common H&E morphology into that high-value virtual MIF.
And that huge scale, 14,000 patients, is what gives you the statistical power to find these really subtle but clinically critical patterns.
Precisely.
Okay, let's get into the weeds of the mechanism. If H&E is a simple blueprint, how does GigaTIME turn it into this complex 21-layer map of protein activations?
So the core of it is a cross-modal translator built on an architecture called NestedUNet.
NestedUNet.
Yeah. You can think of it like a really sophisticated image filter in two parts. The first part, the encoder, takes the H&E image and sort of squashes it down, condensing all that morphological info, the shape of the nucleus, texture of the cytoplasm into its essential features.
And then the second part.
The decoder. It takes those abstract features and expands them back out, but this time into 21 separate high-resolution images, one for each protein channel.
So it's literally translating the visual language. It sees a certain cell shape in the H&E and basically says, okay, based on my training, that shape and its neighbors means there's a high chance of PD-L1 activation right here.
Exactly that. For every single pixel, it's performing a binary classification.
Yeah.
Is this pixel on or off for this specific protein?
And the 21 proteins cover everything.
They're very comprehensive. You've got checkpoints like PD-1 and PD-L1, markers for T cells, cell proliferation with Ki-67, cell death with caspase-3, macrophage markers like CD68. It's a full picture of the immune environment.
Translation quality is everything, though. How do we know the AI isn't just painting by numbers, creating maps that look pretty but are meaningless?
That's where the validation was incredibly rigorous. They didn't just use one metric, they evaluated it across 3 different levels of detail to make sure the translation was solid from the single pixel all the way up to the entire slide.
Okay, walk us through those 3 levels. What's the most basic test?
First is the pixel level. For this, they use something called the DICE score. It's really asking, did the AI get the exact shape of the protein activation right? It's a segmentation test.
So matching the outlines perfectly.
As close as possible. Then you move up to the cell level, which uses Pearson correlation.
And that's looking at what?
Instead of pixels, it looks at little 8x8 windows, about the size of one cell. It's checking if the AI correctly counted and localized the active cells in that little neighborhood.
So it's not just about the shape, but about getting the right cells in the right places.
Exactly. And finally, you have the slide level. This uses Spearman correlation, and it's inspired by a real clinical metric called the immunoscore. This is the big picture.
This one checks if the overall pattern across the entire slide is correct.
Right, the overall density and distribution, because that's what often matters for the clinical outcome. You really need all 3 to be right shape, cell count, and global pattern to trust the results.
And what's fascinating is how GigaTIME, with its paired training data, stacked up against other models like CycleGAN.
It was a night and day difference. GigaTIME blew CycleGAN out of the water on 15 of the 21 channels.
Is there one specific example that really shows that gap?
The best one is the DAPI channel, which stains the cell nucleus. GigaTIME got a DICE score of 0.72, 72% accurate on the shape. The baseline was just 0.12.
Wow.
And for cell localization, GigaTIME hit a Pearson correlation of 0.59. CycleGAN was at 0.03, which is basically random chance.
So it's clear GigaTIME is learning real biological rules, not just copying visual styles.
Absolutely. And that leads to a really interesting insight. The translation quality wasn't uniform. It was best for nuclear proteins like DPI and the proliferation marker Ki-67.
Why there?
Because the nucleus is a compact, clearly defined structure in the original H&E image. The morphology itself is a very strong signal. In contrast, proteins on the cell surface or in the cytoplasm can be much more diffuse and scattered, which makes them harder to predict from morphology alone.
Okay, let's do a quick recap of this section. GigaTIME uses a NestedUNet to translate cheap H&E morphology into these 21-channel virtual MIF maps.
And its success was validated at the pixel, cell, and slide level, proving it's learning real biology.
With the key insight that it works best on nuclear markers because their shapes are so clear in the source image. So this brings us to the payoff. We've created this massive virtual atlas from 14,000 patients. What did it immediately tell us about cancer that we couldn't see before?
Well, the scale just immediately opened the floodgates for discovery. The analysis uncovered over 1,234, to be exact, statistically significant associations.
Wow.
Linking proteins to biomarkers, staging, survival, things that would have taken decades of traditional lab work.
So let's start at the highest level, the pan-cancer view. What known biology did GigaTIME immediately confirm?
It confirmed the classic link between highly mutated tumors and a strong immune response. Tumors with high mutational burden, or TMBH, and microsatellite instability, MSIH.
The ones that look more foreign to the immune system.
Exactly. They showed strong positive correlations with immune infiltration markers, plasma cells, B cells, macrophages, T helper cells. That's Biology 101 for immuno-oncology, and GigaTIME validated it at an unprecedented scale.
Okay, but the really exciting stuff is usually in the unexpected findings, often the negative correlations. What did it find there?
A really critical one was related to KRAS mutations. Across all cancer types, KRAS mutant tumors were negatively associated with immune infiltration. So less CD3, less CD8, and less PD-L1 expression.
Which suggests what's known as an immune-excluded phenotype.
Precisely. The immune cells might be nearby, but they're being physically kept out of the tumor. That's a huge insight for deciding who might benefit from certain therapies.
And speaking of therapies, what about the big one, PD-L1?
So, first, the virtual PD-L1 activation correlated positively with the clinically reported PD-L1 tests, which is a crucial sanity check.
Right, it has to match reality.
But then, when we compared it to cell dynamics, we found something new. High PD-L1 was negatively associated with proliferation markers like Ki-67 and also apoptosis, or cell death markers.
So what does that suggest?
It suggests that high PD-L1 might be tied to a less active, more dormant tumor state. The immune blockade is effectively keeping things in a kind of stalemate.
That's fascinating. Now let's drill down from that pan-cancer view. The real power of precision medicine, is in the specifics, the cancer types and subtypes.
And this is where it got really interesting, because biology is rarely universal. We found 48 associations that were unique just to lung cancer, for example, but we could go even deeper.
Give us an example of that subtype nuance.
The best one is in lung squamous cell carcinoma, or LUSC. In most cancers, high TMB predicts a strong immune response, right?
Right.
Well, in LUSC, that link was broken. High TMB was not associated with increased immune infiltration.
Whoa, so the rule doesn't apply there.
It doesn't. And that might help explain why that specific group of patients often doesn't respond well to checkpoint inhibitors. The AI found a critical exception to a major rule.
That's a whole new way to stratify patients. Did GigaTIME also connect to physical progression like clinical staging?
It did. When looking at the TNM staging system, we found that the primary tumor size, the T stage, correlated positively with immune checkpoint and infiltration markers.
Meaning as the tumor grows, the immune battle heats up at that site.
Exactly. But interestingly, nodal status, the end stage, whether it's spread to lymph nodes, showed almost no association with those same immune markers.
So the main fight is at the primary tumor, and that's not necessarily reflected in the lymph nodes. What about survival prediction? Did you really need all 21 proteins?
Absolutely. This was one of the most important takeaways. While individual proteins could predict outcomes to some degree, a GigaTIME signature that integrated all 21 channels was demonstrably superior for predicting patient survival.
So it confirms the need to move beyond single marker thinking.
It proves it. The whole is truly greater than the sum of its parts here.
Okay, another quick check. The virtual atlas confirmed knowns like TMB and immune activity, but also found key nuances like the immune exclusion in KRAS tumors and the TMB exception in LUSC.
And that the multiplexed 21 protein signature is essential for robust survival prediction.
We've established all this clinical relevance from counts and densities, but GigaTIME gives you images, these rich visual maps. How can the AI use the organization of those proteins to find even deeper insights?
This is where things get really powerful, because now you can move beyond just counting cells and start analyzing the actual spatial patterns.
And the researchers did this using 3 sophisticated spatial metrics.
That's right. The first is entropy. You could think of entropy as a measure of complexity or messiness. Is the protein activation all clumped together neatly, which is low entropy, or is it scattered everywhere in a disorganized way, which is high entropy?
Okay, so it measures heterogeneity.
Yeah.
What were the other 2?
The others were SNR, signal-to-noise ratio, which is pretty straightforward. How strong is the signal versus the background?
And sharpness.
And sharpness, which assesses how clear the edges and boundaries are. That's really critical when you're trying to understand where the tumor ends and the healthy tissue begins.
And the big aha moment here was that these spatial metrics were often better predictors than just counting the cells.
Much better, in many cases. Entropy was the big winner. It improved the statistical strength of the association in 89 different biomarker pairs. It tells us that how the cells are arranged is often more important than just how many there are.
That's incredible. So from spatial structure to combinatorial analysis, what happens when you start looking at how these 21 proteins work together?
This led to one of the most exciting findings. They looked at pairwise combinations. So if a cell expressed protein A or protein B, it got counted.
What was the star combination?
It was combining virtual CD138, the marker for plasma cells, with virtual CD68, the marker for macrophages. That combination was much more strongly associated with 13 different biomarkers than either protein was by itself.
What's the biological story there? Why is that combination so powerful?
It likely points to a coordinated, active immune attack. The plasma cells are producing antibodies, and those antibodies are, flagging the tumor cells for the macrophages to come in and destroy them.
So you're not just seeing 2 cell types present, you're seeing evidence of them working together.
You're seeing the signature of a functional, organized immune assault. We saw similar synergies with other pairs, too, like combining the immune evasion marker PDL1 with the cell death marker caspase-3. It's all about capturing that interplay.
Last big question. How well does this generalize? The model was trained on one set of data. How did it do on a totally independent data set?
A crucial test. They applied it to over 10,000 tumors from the Cancer Genome Atlas, or TCGA.
And
the concordance was excellent, a Spearman correlation of 0.88 between the two populations. The model works. But, and this is a really important point, the Providence real-world data actually yielded 33% more significant pan-cancer associations than the highly curated TCGA data.
So the messy, diverse real-world data was actually richer.
It was richer. It's a huge endorsement for using these large-scale clinical data sets. The big takeaway from all this modeling is what you could call the coordinated nature of the immune response. The T cells, the B cells, the plasma cells, the macrophages, they all cross-correlate. It proves that effective therapies need to engage the whole system, not just one part of it.
That's a perfect synthesis. GigaTIME is effectively tearing down the data scarcity wall, turning cheap, routine H&E slides into these massive, high-dimensional atlases for discovery.
Exactly. The future of immuno-oncology has to be about these comprehensive, context-aware, multiplexed signatures. GigaTIME shows us a path to get there at population scale.
So here's the final thought for you to take away. We saw that the AI was best at translating nuclear proteins because their shape, their morphology was so clear in the original image. If AI can pull this much information about a cell state just from its shape and its neighborhood, what other expensive biological signals we currently measure with complex chemistry are hiding in plain sight, just waiting to be unlocked in our vast archives of routine, low-cost images?